Good evening, everyone, and welcome back to Health Ed 295, EPI for Public Health Practice. Hopefully, everyone has been staying inside with your family, making sure that they're protected and that you all are doing well. I know it's pretty crazy out there, especially in New York. So, um, before anything, just make sure your health is taken care of, anybody that you're responsible for, and also your family. Um, so, amid all everything that's going on is pretty crazy. We're going to continue online. This is our new normal. I'm available if there are any questions or things about content that we've covered, or things about questions about things that we will cover going forward. Feel free to send me an email. I'll respond back uh, in a fairly quickly or timely manner. So feel free to shoot me emails. Today we're going to be talking about experimental study designs and randomized control trials. Up until this point, the studies that we looked at, and the last time we met, we talked about observational studies. So even with cohort studies, we were observing those individuals and we compared groups based on their exposure or, excuse me, an exposure they may not have had. But then we've kind of finally got to this point where, as investigators, as researchers, we actually are assigning people into a certain group and they will receive some type of experiment, whether that's a medication, whether that's a health education classes or something that we want to test because we hypothesize that by experiencing this new health education literature, or going to these courses, or it could be a new medication that is beneficial compared to people who don't receive it. And so, if we don't think that going in, there's kind of no point of actually rolling out the study. So we kind of gather evidence from previous studies or literature that we have out there on the topic to really help us make an argument for why we should go forward with experimental study, specifically a randomized control trial. So for the learning objectives today, we're going to state how study designs compare with respect to validity of causal inference. And so when we last met, we talked about cohort studies and actually being able to look at causal relationships. We can start to look at that now also in experimental studies, because we're allocating some type of, again, some type of drug, some type of treatment to a group, and another group is not receiving that. And so we're able to actually look to see cause and effect there. We're going to distinguish between control experimental studies and quasi-experimental studies. There's one kind of big difference between them, but they're very similar. We're going to describe the scope of intervention studies, define term controlled clinical trials, and give some examples. Explain the phases in testing of new drugs, or what we call as phase. There's 1, 2, 3, and 4, usually trial of clinical trials. We're going to discuss blinding and crossover in clinical trials, define what is meant by community trials, and then conclude with some ethical considerations when you are carrying out experimental studies with human subjects. So, true experimental studies, you could think about these two factors that must be present in order for them to be considered an experimental study. And that's manipulation of some type of factor. So it could be, we think about control group and, an intervention group. The intervention is going to receive a medication. So that's the manipulation factor. The other group they will not receive the medication at all. And so you want to actually see if by receiving this medication, do you see some type of difference in terms of the outcome of health compared to people who don't receive that. And then there's the randomization of subjects. So this reduces what we call a bias. When we randomize people into groups, we as investigators, or if there's a physician, it reduces that bias of wanting to put someone in the intervention group versus the control group. So an example of that would be, and I haven't seen people actually bias and put people into groups, but having worked on some clinical trials, uh, there were patients in a hospital in this clinical trial. There were patients who would have benefited greatly from being in an intervention group. And so some of the clinicians on that particular study, they really wanted their patient to be in the study, but they were really hoping that they were going to be in an intervention group. And there's usually like some type of simulator. I just push a button and it puts people either in a control group or intervention group. But as much as you want to put them in an intervention group, ultimately you don't want to bias and tank the results of the study, which would, basically that's what will happen. So randomizations helps, in short, with all of that and kind of reducing the bias. Um, it helps us to really look at the true association between risk and outcomes. And randomized control trials are just kind of this the hallmark or what we call our gold standard of experimental studies. So one example, actually, when comparing epidemiologic study data versus clinical trials where they clash, was surrounding this women's health initiative looking at hormone replacement therapy. So epi studies have showed that by using hormone replacement therapy, it was beneficial for coronary heart disease in women. But the clinical trials have failed to really describe that same benefit. Um, and so a large body of epi research basically saw that women who took hormone replacement therapy, although they had protection against coronary heart disease, it increased their risk for breast cancer. So what did we do to resolve this question of whether or not it was beneficial or deleterious or harmful to the health of women by taking hormone replacement therapy? They launched a clinical trial. And so this demonstrated that epi findings on cancer were generally accurate. So the clinical trial data and findings basically align from what the epi studies have been reporting on. Um, and the benefits on cardiovascular disease have been overestimated. So this is where kind of going back to randomized control trials and these experimental studies kind of being why they're the gold standard. Because if you are actually allocating someone in a treatment versus a control group, you're able to have more control over the biases and things that can interfere when you're actually running your statistics. If you're doing that other study could have been a cohort study. And although we look at common exposures and follow people up over time, we're never going to get as close the true association between an exposure and an outcome, or in this case, the allocation of some type of treatment or medication and an outcome, in this case was, cardiovascular disease, than if we do a randomized control trial. And so after this happened, hormone replacement therapy use actually decreased. And so from 40 to 80% afterwards. So this is a figure from your book that just kind of ranks different types of study designs, and we've talked about most of these, not all in detail, but you can see that at the top, the validity ranking, meaning it's like the gold standard or like the best type of study design, would be an experimental study, because as a researcher, you have a lot of control over what's going on in terms of, that these people are receiving this treatment. You didn't pick people into placing them in the groups, and so you can reduce a lot of kind of this bias that would interfere with the interpretation of your results. And it kind of goes down. I mean, randomized controlled trials are a type of experimental study design, so you can almost lump those together. Then we got a quasi-experimental, which we'll talk about in a second. Goes down to cohort study. So you can see basically after quasi-experimental study designs, cohort studies become what we talked about last week is like the next best thing that we can get. And we didn't talk about anecdotal or case studies, but you can see ecologic study, which is kind of sits at the bottom as compared to experimental study in terms of what would be the strongest or most rigorous type of study design. So quasi-experimental studies, and I believe I actually mentioned this last week or a couple weeks ago. They're very similar to an experimental, a true experimental study design, but the difference is actually there is the investigator does not randomize the participants in the study. So there could be multiple reasons why you would do a quasi-experimental study design. In some cases, what's going on now with coronavirus, and I know what happened with Ebola, it might not either be ethical to randomize people. It might be inappropriate to randomize people. And so when you think about times of crises and you're actually starting some type of, experimental study or trying to see if, a new medication or new vaccine is more effective compared to what the standard of care is, you just won't randomize people. And it's ranked slightly below a true experimental study design or a randomized control trial. And so, again, like going back to just a true experimental study design, um, you're really trying to see if by actually allocating some type of treatment uh, provides a true benefit or changes the outcome that you're looking at compared to the people who don't receive that. So again, there's a manipulation factor, which we can just think of as a drug. The treatment group is actually receiving some type of medication, and the control group. When it comes to medication and drugs or clinical trials, usually they'll give someone, you might have heard like a water pill or a placebo pill. So both groups will receive a pill because if you gave one group a pill, if you're in the control group and you didn't get a pill, you actually know that you're not in the experiment. You're not in the experimental group, and so that might change your behavior. Maybe you don't want to necessarily participate. You might start to act different, behave differently, and that will change the results and the interpretation of results from that study. So people in the study, if nothing else, they don't know. We're going to talk about this in terms of blinding. They don't know whether or not they're in the control group or if they're in the treatment group. And it's really up to the investigators to try to do a great job in designing the study so that it's not so obvious that, one group is receiving a pill, another group doesn't receive anything, where you would know that you're not, in the intervention group. So there are two kind of broad categories that we're going to focus on of experimental studies. The first type are clinical trials, and so these focus on the individual. So just going back to that example of, I want to see if this medication or this new vaccine is more effective than what we're currently doing now to treat, X. And so you're actually giving an individual something. Versus a community trial, it focuses on the overall community, or they're the intervention versus an individual. Um, and so it's important to note that control trials may be conducted both at the individual and community level. So it kind of really depends on, you might, for example, it'll focus on the individuals, but among people in a very specific community. So it's a community trial that's actually looking at individuals, which that might sound a little confusing, but you can almost think about, again, like if with Ebola, and they wanted to look at specific communities because they were actually impacted by Ebola. So they wouldn't, enroll people in that study at that particular time who were in the United States. So we're going to shift to kind of defining clinical trials. Um, so these are research studies that involve the administration of some type of medication to evaluate its efficacy and safety. So this becomes really important in terms of you thinking about not only looking at whether or not it works, but if it's safe. And that's really a roadblock in a lot of, or has been a roadblock block in a lot of advancements we've made in science and the medical field, just because um, you don't want to give someone a medication that's going to cure X if it's going to kill them in the process. That wouldn't be helpful for anyone. And so, this is kind of the reason why we actually have clinical trials to really test that out to see at what dose is effective and also safe for individuals to take so that we can cure or treat a particular disease. Um, and there's a wide variety of how they'll be used. So the first use was for human subjects without any type of control treatment. But now it's more so refers to the random allocation of tests and control treatment. So, a lot of just science or just any field, it's lessons learned over time. You understand how, altering the design or how that can impact your results or your interpretation of those results. So some characteristics of clinical trials, they're supposed to be, very thoughtfully planned and rigidly enforced. The protocol is another big thing. It's up to, and it's kind of, I spoke about this with the cohort studies or any type of study. The data that you have from your study are only as good as the people who are running your study, the people who are, the research assistants, the people who are looking at the data, because, you know, if you as an interviewer, and I have this happen not to me directly, but when I worked in studies where I had to interview participants, if you start deviating from the basically the protocol or the material you're reading from and like actually doing the interview, it might change the answers that those participants give you. So it's important to train and retrain staff to make sure that everything is going the same. Um, they're tightly controlled in terms of who's eligible, who's delivering the intervention, and in terms of monitoring the outcomes. So usually for clinical trials, they're very specific eligibility criteria. I mean, because they don't want, again, to kind of use an example of, excuse me, if we were looking at Ebola and there was no cases of Ebola in the United States, you wouldn't enroll someone in the United States because, or someone who didn't have Ebola at all, I guess. The duration ranges, so some can range from some days to years. Participation, again, is only usually limited to a very specific group of individuals. So once the participants or subjects, human subjects, agree to participate, they're randomly assigned into one of the groups. So either the intervention or control group. And there are different ways that you can randomize, but from my knowledge, this is usually done on a computer to just reduce that urge as a researcher to want to put someone in an intervention group because it's going to be beneficial for them. And it's hard. I'm not a clinician, but I know that I work with patients, and there were some instances where like this person being in the intervention group is gonna be beneficial to this person because if they're in a control group, they're not gonna get really a benefit from the study, I guess, which sounds kind of bad, but just in terms of science and reducing this bias, you can't just put people into groups because that's gonna again taint the data that you get and ultimately the analysis and interpretation from the study. So they're prophylactic and therapeutic clinical trials, whereas prophylactics trials are looking to see if you allocate some type of treatment or drug, if it's going to prevent someone from getting disease. Versus therapeutic, you can think about it as a treatment. So this is some type of surgical procedure or some type of drug. Think about we developed some type of new or well, it wouldn't be a vaccine, but some type of medication that we want to test to see if it's more effective of treating a particular, cancer, for example. So it would be like more of a therapeutic clinical trial. So in terms of the closure of clinical trials, they're referred to as the clinical endpoints. And this may include rates of disease, death, or recovery. The outcome of interest is measured in the intervention and control arms of the trial to evaluate efficacy. And so another reason why you might actually close out clinical trials actually goes back to if we are giving someone a medication, for example, and just it could be cancer. We develop a new medication to treat cancer, and we find that after a month, and you have to report these events. After a month, we noticed that 5 patients all of a sudden started to develop hypertension, and they didn't have hypertension before. Once that happens, it's your responsibility as a researcher to look into that because it might not be random. It might be some type of adverse effect that you are inducing based on providing people this intervention or this medication. Um, let alone if people start dying or something like that. So it becomes really, really important, and clinical trials are tightly regulated for that purpose. If anything kind of happens, you are legally responsible for reporting that. And by failing to do so, I know that you can lose. The study is definitely going to be cut, but you can actually attack. That taps into, your job security of your clinician. That can tap into your licensing. So it becomes really important. So, a couple examples of some clinical trials. The first is a Medical Research Council vitamin study, and they wanted to look at the relationship between folic acid and preventing neural tube defects. Well, as it comes or finds out, this is now like kind of the standard. When I learned nutrition, it was I just knew about folic acid and spina bifida, like that was like a thing. But at one point, they developed this study to really see if supplementing folic acid among pregnant women actually reduce the risk of spina bifida or neural tube defects. Another is the South Bronx um, SCD program to look to see or evaluate the effectiveness of education efforts in order to prevent the spread of STDs. There are different type of. I mean, when we get into we don't really talk about STIs so much here, but there are different type of efforts or different type of trials that have been rolled out to see if they reduce the risk for a particular STI or try to reduce the spread and transmission of STIs. So now we go back to blinding, or some might refer to it as masking. And this is, basically you could think about, again, if people enrolled in a study and we randomize them into a group, um, you don't want people to really know which group they're in because they might start to behave differently, or they might decide they don't want to participate at all in the study. And this could be, it can range from if you're getting, additional health education material um, in addition to what's kind of the standard of care, versus if you are a cancer patient and you're enrolled in a trial potentially for this new treatment, and so you might not actually receive the actual new treatment that we're actually testing. A lot of people might feel a certain type of way about that, so blinding becomes important. And it's also important to let people know that they could be in either group as well, in the informed consent that they provide, and not be misleading with that. And so there are these three kind of levels of blinding that we can do. We can do the single blinding, in which the case, the person who's in the study, they don't know which group they're in, but the researcher or the investigator and the people who are analyzing data, they know who's in which group. We have double blinding, which is usually more kind of like the standard, where both the researcher and the people who are in the study, neither of them know which group that the individual is in. Again, because you don't want to start treating a person differently as a clinician or as a researcher, and you don't want the person and the participant to start behaving differently either. Um, then there's this third level of blinding, which occurs sometimes, is where not only you have the participants blinded, you have the researchers blinded, but you also have the person who's analyzing your data blinded. They don't know who's in which group. They just run their analysis. This in an ideal world, that would be kind of the best thing to do. There are reasons why you would not do that. But you could think about it. If the person who's analyzing your data, they work on your research team, more than likely everyone on the research team want basically the results and analysis of those results to say, yes, by allocating this medication, we saw that, you know, um, people in this did not have this outcome as much compared to people who did not receive this medication. That's what you want to find. But if you don't find that, there are little ways that you can tweak the data, or you can run a certain type of analysis and you can end up finding some type of statistically significant result. And so in order to kind of reduce the urge to do that, some people will do this triple blinding, this extra layer of blinding or masking. So your book kind of talks about these four different phases of clinical trials. I just want you to know that the phases exist, and kind of how new medications or new treatments are developed through clinical trials. They go through a series of these different phases. Um, and so it's basically this lengthy process helps to protect the public from a potentially harmful or deleterious vaccine, and to satisfy urgent needs for new vaccines. Right now, what's going on with coronavirus? They are developing, or COVID-19, I should say more specifically, they're developing a new vaccine, and I actually saw a call for they're trying to basically get people, or they're looking for volunteers to enroll in a clinical trial for COVID vaccine, COVID-19 vaccine development. But even if they develop or start to develop that, it has to go through all these phases before we can put this out in the public. Because, and I'm sure everyone who's going to be a candidate for that particular study, they have to probably be very healthy within a certain age. They can't basically fall into any of those groups for what are considered vulnerable groups for COVID-19 right now, just because they don't want to have any adverse effects from the vaccine itself. And so that's why it takes a long time to develop. And I won't even go into it of why, it's not true that the vaccine is going to be here in a week or so. It takes a lot longer than that. So I talked about randomizing subjects, putting people either in an intervention or control group. Um, but there could be instances, which I'm going to kind of talk about in a second, where you actually, someone might be in an intervention group at one part of the study, and then you move them into the control group or vice versa. So that's referred to as crossover study designs or within clinical trial. And this is when any change of treatment for a patient in a clinical trial switches during study treatment. So in planned crossovers, a protocol is developed in advance, and the patient may serve as his or her own control. Unplanned crossovers also happen for various reasons. And sometimes it could be that a patient was receiving a medication and they started having severe adverse effects or harmful effects, or that was specific to that individual. And so they might be swapped into the actual control group at that time. Or they can just decide they don't want to participate in the study at all. So going back to ethics, and we have a whole, at the end, I think it's the very last class we talk about ethics, because it's really important in any type of research. And there have been groups who have been abused and things that have been very unethical that have been researched. So it's important that everyone's aware of what those really bad studies that happen in the past, what they are, and how you can be ethical in terms of the research you do or the work that you do involving human subjects. So really important. The benefits of the study must outweigh the risks. So and there's an example at the end. It's actually one of the papers in the next journal summary that we're going to talk about. If you might find a minimal effect from actually giving someone a new medication for cancer treatment, for example, but in the process, you're going to destroy their liver or something like that, or the people are going to go into kidney failure. There's no way that you would be able to start this trial, whether that's for the trial in general, or maybe it may be for that individual. And that's why it's important to have exclusion criteria in the study. That person might otherwise be an eligible candidate, and if liver function was a potential risk, you would want to assess that person's just liver or history of different things that might, like alcohol consumption or other things that might have affected their liver in the past, because that will be considered a very high risk individual by enrolling them in the study, and the benefit might be minimal. Other ethical issues include informed consent. So you can't enroll anyone in the study without their knowledge. That will be considered highly unethical, and and they're different. It's not only just asking someone for their permission. It's actually when you design or write informed consent forms, it have to be written in the language that person speaks. It has to be written at an 8th grade reading level. Like, there have to be things in place, because you can get someone to agree to something and the words that you actually just spoke to them, or even what they read, just went over their head, and they may not have any questions and just enroll just because, again, there's this power dynamic sometimes with clinicians or researchers compared to participants or patients, and so you just want to reduce that from happening. Um, Other ethical issues involved with withholding treatment known to be effective for individual. This is where it becomes really, really tricky in terms of a clinical trial involving some type of medication. Um, at the very minimum, you have to give that person what's considered, or the people in the control group would have to receive what's considered the standard of care of how other people are being treated for that particular outcome. You can't not give them anything at all and give the people in the treatment group some new medication. That would be very unethical. Um, you have to monitor the side effects. So again, you're gonna know that there are certain side effects from a medication in a clinical trial, but you need to just monitor, get labs ordered, frequently. And this is the researcher, in terms of designing the study. They need to have protocols in place to kind of keep these things on track. Uh, you have to protect the interests of the individual and their privacy becomes important, and decide when it's appropriate to withdraw a patient. And again, this is going to be up to you as a researcher, clinician, whichever your role is going to be on that particular study, of kind of just monitoring participants and seeing how things are going over time while they're in the study. So in terms of reporting of clinical trials, we use what's called a consort statement. And this is basically a checklist to help us actually make sure that we have everything that is supposed to be there when we're actually submitting a manuscript for a peer review journal. And so what you all are actually going to do, there's an assignment that does something similarly. They're called the Strobe guidelines. And really the big difference between Strobe and the consort is that Strobe looks at it's reporting for observational studies, whereas consort is reporting for randomized trials, experimental studies. So in terms of summarizing clinical trials, some strengths, they provide the greatest control over the amount of exposure, the timing and frequency of exposure, and the period of observation. So they're very tightly kind of controlled by, that's why they're called randomized control trials. People are randomized and it's tightly controlled. And so you can really look at cause, effect, and causation. And this is really why the, randomized control trials are considered kind of this gold standard or the best type of research you can do. However, there are always limitations. So limitations include it's an artificial setting. So because it's so tightly regulated, you might be having people do behaviors that they normally may not do, or you might influence something that wouldn't apply in the real world. Limited scope of potential impact. So again, it's usually for a specific subgroup of people, and it's not necessarily going to be generalizable to everyone, and that's okay because it might be a very specific question that you're looking to answer. Adhering to protocol can be difficult to enforce. So the longer the trial takes place, getting people to continue taking medication, sometimes all medication is going to have some type of side effect, that can be difficult. And then again, kind of going back to those ethical concerns that I talked about before. So kind of in the end, kind of concluding or getting closer to the end, we're going to shift to talk about community trials. So you could think about those clinical trials taking place with the individual, whereas the community trials are going to involve the entire community. And so community intervention trials try to determine the potential benefit of new policies and programs. And so for the intervention, it could be any program or other planned effort designed to produce some type of change in a target population. And in this case, the community could be, a city, a county, a state, or a school district. One example I can kind of provide of a community trial to put, I know concrete examples are better, that I was involved in was a workplace exercise program in order to try to reduce obesity and cardiovascular disease and diabetes. And so basically the target was actually the state or local, state, and city public health departments in Los Angeles. And so there was a designated people who were trained to lead kind of these exercise breaks that the job gave them. So in addition to kind of having your lunch break, you also got a 30-minute exercise break that anyone in that job could take. And they also started stocking the vending machines with healthy snack options. And they removed kind of, I think, soda and sugary beverages out. And so they wanted to actually look at that community to see if by implementing or having this type of program, will we see shifts in the health or behaviors of individuals in that particular workplace. So similar to any type of study, you want to determine which communities will be eligible and willing to participate. You want to collect some information at baseline or at the beginning to be able to measure the problem. So it's important to kind of just going back to when we're thinking about cohort studies, you at least have that baseline or beginning point and at least one observation after that, because otherwise, if you didn't record what happened at baseline or at the beginning, you don't have anything to compare the next observation point to. And it can be a variety of measures that you're looking at: disease rates, knowledge, attitudes, and practices. Communities are randomized and followed over time, and the outcomes and interests are measured. So in terms of advantages, they represent the only way to estimate directly the impact of change in behavior or modifiable exposure on the incidence of disease. And so kind of giving that same example of that community trial that happened at Los Angeles Public Health Department workplaces. You were looking at a very specific type of group of people, or workplace, or really government employees working in public health, to see if this trial was going to actually be beneficial. Then they might start to actually expand that out to look at different government employees, not outside of health, and then eventually look at different type of workplace environments to see if it's effective. So in terms of disadvantages, they're inferior to clinical trial in respect to your ability to control for interest into the study, delivery of intervention, and monitoring outcomes. So you're looking at the entire workplace with that example I gave. You're not necessarily looking at individuals so much. And so, you have less control in that sense. There are fewer study units. So capable of being randomized, which affects your comparison that you're making in community trials. And they're affected by population dynamics, secular trends, and non-intervention influences. People might decide they're going to quit that job, or 5 more people might get hired, and so that different things that happening. I mean, if you were doing a trial right now, what's going on? That would research just stop, but there are going to be some people that might not want to participate at all, even once things kind of get back and going. And then kind of following up in terms of how we evaluate this. So there are these 4 different steps in terms of evaluation, or how we can evaluate whether or not what we're looking at the trial, if it was effective. So starting with the formative evaluation, did all the plans work as we basically thought that they would work? The question you're looking at in terms of the process evaluation, you're looking at the process itself. Is the program serving the top target group? Did it work as planned? The people who are implementing it, how did that work out? Because you might have a great trial that you're starting, but the people who are responsible for implementing it at that particular workplace, for example, they might be bombarded with trying to do their job and also being the only point of contact for this at their study at their workplace. So you want to measure to see how that went as well. The impact evaluation. So did the program produce any type of changes in the target population? Did the workplace changes to vending machines and also giving these short exercise breaks, that we start to see that impacted that workplace or the health of those people who work there? And then the outcome, did the program accomplish what the ultimate goal was? And so usually with the evaluation of clinical trials or these community trials, you're looking at, you're evaluating at each step. You want to understand all the information, because that's going to help you determine whether or not only was it effective, but if it wasn't effective, maybe why was it not effective? Was it the process? Again, like that person is bombarded by being a point of contact at that workplace and also trying to do their job. Maybe there should have been 10 people designated instead of one. And so you want to kind of really understand the process, how it impacted your outcomes. So just summarizing just some big key points for experimental studies and what we talked about today. Experimental studies involve the manipulation of some type of factor and a randomization of subjects. Randomized controlled trials are considered the gold standard when it comes to experimental studies and generally for any type of study design. So if anybody ever asks you, if you don't remember any type of experimental study design or what's the best type of study, your answer could always be randomized controlled trials, because those are considered kind of the best that you can get in terms of study designs. Right below that are quasi-experimental studies, which are very similar to experimental study design, but remember that you don't randomize the participants. However, people do receive a treatment or they're in a control group. And then ethical concerns must be carefully considered before you even start a study, but also while the study is going on. There have to be benefits outweighing the potential risks. So this was our last study design that we're going to cover this semester in the class. We're going to move on next week to talk, well, it's actually in a couple weeks, of talking about measures of effect and then different kind of subgroups of epidemiology. So social epidemiology, molecular and genetic epidemiology, those other little subgroups of, basically eventually as a epidemiologist, you actually develop a niche in a specific area and you kind of have that expertise. Again, so hopefully everyone's taking care of themselves, taking care of their families, making sure that you all have food and everything you need. Make sure your mental health is protected. Social distancing doesn't mean that you can't communicate with people. Feel free again to reach out to me if there are any questions or concerns that you have via email. All right.